Lauren actually asked me to talk a little bit about clinical research. And in your handouts are some slides and tables of some specific drugs. But I'm not going to dwell on the specific drugs. I think it's better to get a bigger picture. And frankly, what I'm going to talk about is sorts of nuts and bolts of clinical research, how we do trials, a little bit about the standard drugs, some of which are still good drugs, and then give Pam the luxury of talking about the more exciting things that are going on in the newer drugs that we're all hopeful will have an impact on some patients with neuroendocrine tumors. Okay? And I think we're just going to get started, because the sooner we get started, the sooner everyone gets out and gets fresh air on a nice day. So I'm going to start with sort of describing how we test drugs. Drugs don't come out of the air. They're not just given to us by Genentech or Novartis. These are drugs that have to pass certain hurdles in the laboratory. They might be drugs that are specifically designed to attack a certain pathway that we think might be important for cancer. They might be specifically designed to attack a certain molecule that we know is on cancer cells. Or they may be drugs that just by chance looked like they were pretty good in the laboratory at killing cancer cells, and that they were relatively safe in mice and cured a few mouse tumors, and therefore kind of jump forward to be ready for human studies. So we start with preclinical work, where in the laboratory they show that a drug can work on mouse tumors. Now, one of the problems for neuroendocrine tumors, and one of the problems for the patients, is that we don't have good mouse neuroendocrine tumors. We have tons of mouse breast cancers, tons of mouse pancreas cancers, colon cancers. We've had a rough time growing mouse neuroendocrine tumors. There are one or two, but we wonder whether they're even valid. So we need better tumor models. So that's one of the areas of investigation. Once we have demonstrated that a drug looks promising enough to try in patients, then we do what we call a phase 1 trial. Now, there are a lot of misconceptions about phase 1 trials. Phase 1 trials, the scientific endpoint is to establish safety. But establishing safety is not really how it's interpreted. It's defining toxicity. So, of course, if we're given a new drug, we have to make sure that we know, well, what are the side effects? How is it metabolized by the body? How long does it last in the body? Does it have any effects on the kidney or the liver? Does it have any effects on the person in terms of diarrhea, hair loss, numbness, tingling, nausea, any of those side effects that many of you know all too well? But, so the scientific endpoint is just how does the body handle that drug? But that doesn't mean we can't hope for efficacy, because these are drugs, after all, that cured a few mouse tumors. We're hoping that they can have a role in patients too. So we have phase 1 trials where we're testing drugs in people. Because they're phase 1, because the scientific endpoint isn't what percentage of patients with colon cancer have a response, what percentage of patients with neuroendocrine tumors are cured. Those are nice endpoints, but the scientific endpoint is safety and tolerability. And therefore, any patient would be eligible for a phase 1 study. So if you go through the NCI.gov list of clinical trials, you won't find a whole lot of clinical trials for neuroendocrine tumors, but you'll find a lot of phase 1 trials where neuroendocrine tumors would be eligible potentially. And again, the scientific endpoint is safety. Anyone with a measurable tumor is usually anyone who's in good shape with a measurable tumor could be eligible, and you can get a phase 1 drug, or a combination of drugs. Phase 1 trials are usually small, 15 to 25 patients or so. And again, it's to define side effects and try to determine what's the best dose of the drug. Well, if the tumor passes the mouse test and is determined to be safe at a reasonable dose in phase 1, the next question is, how effective is it? And then we have to take a guess. We have to say, well, let's test this in colon cancer. Let's test this in breast cancer. Let's test this in lung cancer. Do you know why they test those 3 cancers? It's a big market. I mean, number 1, it's a big market. Number 2, they're common. The fact that they're common means that you can test them quickly, which is not, it's one of the problems is how long it takes to do clinical trials. So you don't get too many drugs tested first in neuroendocrine tumors, although that may be changing soon. So the question then is, how effective is it in a defined population of patients? So you define a cancer. So most phase 2 studies are disease specific. Okay, phase 2 study is asking the question, how effective is this drug for colon cancer? How effective is this drug for pancreatic neuroendocrine tumor? How effective is this drug for carcinoid tumors? Phase 2 studies are usually limited to 25 to 50 patients or so. And we just get a kind of a ballpark estimate of that. If we think that the 25 or 50 patients here did okay, we think that the drug looks reasonably effective, then the next phase of study is to say, well, how does that compare with the standard treatment? So we've already established safety. We've established that it does work for some patients. We think it works for enough patients to take it to the next step. And this is the one the FDA likes. It's a phase 3 study. It sometimes gets bad press, but it's a randomized study. It takes patients with the same disease. If it was colon cancer here, it would be colon cancer here. Breast cancer here, it would be breast cancer here. If it was neuroendocrine tumor here, it would be neuroendocrine tumor here. In the phase 3 study, it simply asks the question, how does it compare to a standard treatment? Now, it may be that the placebo is a control, which is to say that if you have a disease for which there is no standard treatment, then it's perfectly ethical to randomize between drug X and a placebo. If you have a disease with an accepted standard of care, then oftentimes what we'll do is we'll say, what about the standard of care plus placebo versus the standard of care plus drug X? And then at the end of, well, this study might take 200, 500 patients. You can see that that's a limitation already when you saw the numbers that Dr. Visser was showing you of surgery studies for pancreas, for neuroendocrine tumors with 20 or 30 patients. It could take 200 to 500 patients to do an adequately powered, statistically significant phase 3 study. Now, what I didn't put up here is time. This study might take 6 months. It could take a year and a half. This study, depending on how many centers participate, it could take a year or it could take 3 years. This could take 3 to 10 years to do. You add them all up, it's maybe $20, $30 million. And then ultimately, if this one's positive, the FDA says thumbs up. Okay? So that's the process that we go through in clinical research to try to get a drug approved. I'd be interested in Dan's process for getting procedures approved is much shorter than this, but this is what it is for drugs. Now, there are important things to recognize when we look at studies, because the ultimate outcome of the study depends on the design of the study, and so we have to really be careful with our interpretation. I'm sorry, this kind of got scattered when I went from Mac to PC. So let's think about eligibility criteria, meaning we have to define a population of patients and then decide, well, this is the group we're going to treat. And so the outcome might only reflect that group of patients. But maybe we can extend the interpretation of that outcome to maybe affect other groups of patients that were not actually studied in that trial. But you always have to remember that there's a defined group of patients for which that treatment was offered. And it might not affect you or the person sitting next to you because it might be a slightly different disease or a slightly different population. So we had a nice talk this morning from Terry Longacre talking about the neuroendocrine tumor types with well-differentiated, poorly differentiated. She called them grades, grade 1, grade 2, grade 3. Those are 3 different, biologically different diseases. And well-differentiated can behave very differently from poorly differentiated tumors. In fact, when we talk about neuroendocrine tumors for the purposes of this audience, we don't even include these. That's a tough crowd. The well-differentiated or moderately differentiated or grade 1 and grade 2 are the ones that we usually talk about. We also talk about the site of origin. Do you just combine all neuroendocrine tumors? And because they're so rare, it makes sense to, we'll just take any comers with neuroendocrine tumors so we can get our 50 patients and publish our results. But is it fair to combine apples, oranges, bananas, all kinds of different fruit, when in fact there may be significant differences among neuroendocrine tumors depending on where they arose, whether they arose in the stomach, the colon, the small intestine, the appendix, the pancreas. One really key part that's becoming more and more important is, was that tumor growing? Which sounds silly. Tumors grow. Some patients with neuroendocrine tumors grow so slowly that it might be years before you could even see it on a scan. And you can follow it on a scan, just follow a millimeter at a time, and I've had patients I've taken care of for 10 years and never treated them. Never treated them with anything. And yes, if I look at from one scan to the next every year, there's no change. If I look this year's scan back 10 years ago, it might be this much different. So that's slow growth. Do those people need to have these same treatments? Probably not. But some trials don't stratify based or don't discriminate based on whether the tumor is growing or not. Most of the new ones do. Most of the new ones require that the tumor has demonstrated some degree of growth in order to be eligible for the clinical trial. Prior treatment or not. Some studies will not allow you to be on the trial if you've already been treated. Or some studies will not allow you to be on the trial if you've been treated with a certain type of drug, because they're testing a similar type of drug, and they don't want to compromise the results by accepting people who might already be resistant to that drug or that class of drugs. Okay? So these are really important eligibility criteria that are not always all that well spelled out in certain studies. And part of our work on the NCI task force is to try to get all investigators around the country, including Emily Bergsland at UCSF, who's part of this task force, to accept the same eligibility criteria so that we can start comparing trials between here and Harvard. Measure of success. How do we measure success? It'd be nice to say we measure success in terms of, well, what percentage of patients are cured between drug A and B. Well, cure is a forever word. And it's not quite right that oncologists have trouble saying cure. It just doesn't roll off my tongue as often as I'd like it to. Cure is a forever word. We like remissions. So what's a remission? A remission means that the cancer's disappeared from view. So a complete response is a remission. That's wonderful. We can no longer see the cancer. A complete response that lasts forever is a cure. But that's why we don't talk about cures right up front. We're aiming for remissions. A partial remission means that you've shrunk the tumor quite a bit. And we have academic definitions that Pam might get into a little bit later as to how we define a partial remission. And then stable disease. Well, if this is pancreatic adenocarcinoma and I had a drug that caused stable disease for 6 months, that'd be a blockbuster drug. But if it's pancreatic islet cell tumor and I kept the disease stable for 6 months, the FDA would yawn. They'd say, well, that's no big deal because most of those patients have stable disease for 6 months. So it's highly dependent upon the subset of patients that you pick for the study. If you pick patients with growing disease, then this might be a very important finding. And that's what we're tending to do now. Naturally, if you have progressive disease, you know that that drug didn't work because the disease grew despite it. Well, these are measures of shrinkage or growth of the tumor. That's what response rate means. These are measures of time before the tumor gets worse. Progression-free survival. Progression-free survival means how long does that person survive? That doesn't mean how long the person will live. How long a time will that person go before there's evidence on a scan that the tumor has progressed? Okay? Very simple concept, but hard to measure, particularly for slow-growing tumors. You need a longer time to watch for a slow-growing tumor. Okay? Overall survival. That's an easy one to measure. You're either alive or you're not. And if you're not, that's a data point. If you are, you're a data point. It's binary, right? That's the gold standard. For neuroendocrine tumors, it's very hard to do that because we've got to wait 15 years to do the study. That $20 million study now costs $50 million because I've got to employ data managers longer than my career will last, because you have to study people over a long time if you do that. Now, for pancreatic adenocarcinoma, we can get those results in a matter of just a couple years because the survival is so poor. So it all depends on the type of tumor that you're measuring. Okay, so now I'm going to talk a little bit about therapeutic options, and then we'll have sort of a panel discussion afterwards to talk about this too. We heard from Dr. Katz-Nelson about drugs for carcinoid syndrome. That's for the secretory function of the carcinoid tumors that causes either flushing or diarrhea or other problems. He talked about Cushing's disease as one rare manifestation of carcinoid. The peptide receptor radiation therapy, I'll just have one slide, and I'm happy to answer questions. It's coming to the States, but like at a glacial pace. Liver-specific therapies, Dr. Z is going to talk about. Pam's going to talk about these, and I'm stuck with the really boring stuff. So let me go through the boring stuff. So somatostatin receptor. Somatostatin is octreotide. It's kind of the endogenous octreotide. Octreotide is just a slightly modified somatostatin that makes it a better drug. In the cell, if you imagine that this is a tumor cell, a carcinoid tumor cell, actually has 5 somatostatin receptors. Anything that works well in biology often occurs in families, and there are families of drugs. So if you have a signaling mechanism that works well, biology will make a family, and each one of these might have a slightly different function. But each one of these can bind to somatostatin, or octreotide, or lanreotide, or pasreotide that was already discussed. Now there are 5 somatostatin receptors. 80% of neuroendocrine tumors have a lot of SSR2, so the somatostatin receptor 2, and then a few, there are a few others that are further down the list. And octreotide has its highest affinity for the one that's most commonly on neuroendocrine tumors, so no surprise that it's been picked for that. Now, the anti-secretory function is an SS2, so that's why octreotide is a good drug to cut down diarrhea in patients with neuroendocrine tumors. These are the 5 somatostatin receptors, and it turns out, though, that those same receptors can sometimes have an effect on the ability of the cell or the tumor to recruit blood vessels, and so it can be angiogenic or anti-angiogenic, proliferative or anti-proliferative, induction of apoptosis. This is a term called a biological term for suicide. If you give these drugs, you can sometimes convince the cell to commit suicide. That's nice. It turns out that cells commit suicide when they feel like something's gone wrong. And if you tickle a pathway that convinces the cell that they've gone the wrong direction, they can commit suicide. So one treatment for neuroendocrine tumors, surprisingly, is octreotide. We just talked about octreotide for the carcinoid syndrome. Now we're going to talk about the tumor growth. And so Lauren asked me to define what PROMID was. So some people who have carcinoid tumor have heard of the PROMID trial. This is what PROMID is. PROMID is placebo-controlled double-blind perspective, randomized study of the effects of octreotide on the control of tumor growth in patients with mid-gut tumors. So it's a study of mid-gut carcinoid tumors, that means small intestine and appendix, in which patients received either placebo or they received octreotide LAR, and then tumors were measured over time. Okay? So that's a study that was just presented by the German group last year. So this is a study, 85 patients, all well-differentiated mid-gut carcinoids or neuroendocrine tumors, randomized, half got placebo, a shot in the butt, but it was a saline, and half got a shot in the butt with octreotide. And then the question was, what was the rate at which patients eventually had tumor progression? And so if you hear the proportion without tumor progression over time in months, couldn't see the numbers. Here's the placebo, and here's the octreotide arm. And so if you look at the time to median progression, where 50% of patients had some degree of progression, for the placebo it was 6 months, and for the LAR, for the octreotide, it was 14 months. So a fairly significant finding in terms of delaying ultimate growth of the tumor. It didn't say that tumor shrunk. It didn't say anyone was cured. In fact, the overall survival was the same, whether you got the octreotide or placebo. But again, you don't expect people to die from this disease in this time frame. This is a disease that grows slowly, but it looked like it grew more slowly on octreotide. Okay? So that's the PROMID study. Now, it's not a perfect study. It's not a perfect study. It's still small numbers. I showed you 85 patients. I told you on the previous slide that for a phase 3 study, we want 200 to 500 patients. So it's not a lot of patients. They didn't control for those tumors that were growing or not. So maybe on the control arm, they had more tumors that just weren't growing. We don't know. They didn't measure that prior to starting the study. There was no control for carcinoid syndrome, which is a little bit unusual. There were no responses seen. There's no shrinkage. Whether you got placebo or the octreotide, you didn't shrink tumors. And curiously, there wasn't any benefit observed in the bulky disease. So you took the people with the largest tumors out. They didn't seem to have any benefit. So still some questions about the study. What are the implications for practice? Well, I think since I have a number of patients who've been out 15 years without a single drug, without any growth of disease, I could have given them octreotide, a shot in the butt, every month for those 15 years, but they certainly didn't need it. And so I think it's reasonable to wait for some degree of growth and then go ahead and offer the octreotide. Lifetime of therapy, when do you stop? We don't know. There's nothing to measure. You stop, I suppose, if the tumor gets worse, but certainly it's not going to shrink the tumor, so there's no way of knowing when you should stop. Should we give it with combination with chemotherapy? We don't know. People are testing that now. Should we apply this midgut study to other neuroendocrine tumors? We don't know that either. So those are kind of the open questions for researchers to try to sort out for us. Now I'm going to talk just briefly about chemotherapy for neuroendocrine tumors. Again, which neuroendocrine tumors? The well-differentiated, so we're not going to deal with the poorly differentiated. That's a completely different type of disease. We're going to talk about pancreatic versus carcinoid, and there are a variety of carcinoids that you heard about this morning. And I'll just touch a little bit on the bronchial lung carcinoid. Which chemotherapy drugs? I'm actually not going to go into much detail. I'm going to skip through a few slides so that Pam can show more interesting data. But some old drugs, some newer versions of old drugs. DTIC we've had for 30 years, temozolomide for about 8 years. It's a different version, an oral version of an old IV drug. Capecitabine, 5FU we've had for 50 years. Capecitabine we've had for 15 years. And then doxorubicin, some platinum drugs. Okay. So, what about the basics? Why are so many of you in this room with a tumor for perhaps a number of years, and yet you still have your hair? Or you haven't had real chemotherapy. So if you know somebody with breast cancer, they say, well, geez, you haven't had real chemotherapy. Or your blood counts are still good. Why haven't you, why is that? Well, because chemotherapy can work for this disease, but it doesn't work great. It has some activity. It can shrink some tumors, not as many as we'd like. Whenever it does have activity, it seems to have more activity for the pancreatic neuroendocrine tumors than the midgut carcinoids. So midgut carcinoids are tougher to shrink than the pancreatic neuroendocrine tumors. The response rate and the duration of response, how long it controls disease, highly variable from study to study, highly variable. And there's no overwhelming sense of survival advantage. At least we don't have those kinds of trials that would demonstrate, aha, you definitely need this chemotherapy because of the stunning survival advantage. And therefore, there's not a whole lot truly approved by the FDA. And also, because there's not a whole lot truly approved, there's no true standard therapy. It allows companies to do a placebo-based control for these diseases. Because there's no standard therapy, so if there's no standard therapy, you can have a placebo versus a new drug. Streptozotocin, I won't really talk about. It was first thought to be an antibiotic, and subsequently is found to be toxic to islet cells. Islet cells are the cells that make some hormones in the pancreas and the source of islet cell tumors. So if it kills some islet cells, maybe it would be helpful for pancreatic cancers. And the FDA actually approved this drug. I think this is the last drug approved by the FDA for neuroendocrine tumors, 1976. Okay, so streptozotocin, I won't go into any details except to say that response rate, you have to read the fine print. Because these responses, which look pretty good from 1992, these responses included a physician who would feel the liver and say, well, that's better. Honestly, that's one way of measuring it. And others were just chemical responses, meaning the blood markers got improved a little bit, and that qualified for a response. More recent studies required actual shrinkage to a certain degree. So that's why it's not that we've gotten worse, we've just gotten more stringent in how we define response. Okay? I'm not going to go into detail here except to say that DTIC is an old drug, temozolomide is a new pill. They both seem to have some benefit, always more so in pancreatic neuroendocrine than carcinoid, but some benefit in carcinoid. And one of the clues as to why that might be is that temozolomide is a drug which binds to DNA. That's the problem. Most of our chemotherapy drugs are random damaging agents for DNA. It's only the PAMS drugs, the targeted drugs, that are new and more exciting and more specific. These are random drugs that damage DNA. So you bind DNA, and when DNA tries to make itself over again, it screws up because there's this lump here. And it makes a mutation, the cell might die. So there's an enzyme called MGMT that can repair this lump. It can recognize temozolomide bound to the DNA, and it just gets rid of it. So it just cleaves it off, makes a new strand, fills in the gap, and now you have nice DNA, and the tumor cell survives. Okay, so cells that have this repair enzyme can repair the damage to temozolomide. Cells that don't have the repair enzyme, they can't. They try to divide. When they divide, they end up screwing up their DNA, and the cell has a sensing system for when the DNA is screwed up, and it commits suicide. Because you don't want to give bad DNA to your daughter cells. So when a cell's ready to divide, it screens the DNA, it looks at the DNA in each daughter cell, and if the daughter cell has lousy DNA, they commit suicide. It's for the good of the organism. Okay? And so that's one mechanism by which temozolomide works. It turns out that most carcinoid tumors have lots of MGMT, but a fair percentage of pancreatic neuroendocrine tumors lack MGMT. And that might be why this may work in pancreatic neuroendocrine tumors. So that needs some more assessment. This is just one of the first experiments showing that for pancreatic neuroendocrine tumor. This is work from Matt Kulke, our colleague at Harvard, who does the same kind of work. He stained for MGMT and stained here for MGMT. There's no MGMT in this. This is all tumor. There's no MGMT in the tumor. There's tons of MGMT in this tumor. And this tumor responded. This tumor did not. And I won't go into the detail of these other things. Oxaliplatin is just one of the platinum drugs that Pam might talk about. Are you going to mention that? Yeah. And so conclusions, challenges from cytotoxic chemotherapy. Number one, cytotoxic chemotherapy can be active. It can shrink some tumors. Again, more so in pancreas. Temozolomide with chemotherapy looks promising, maybe especially so for pancreatic neuroendocrine tumors. The role of MGMT may be important in defining who's going to respond and who's not. So we're trying to measure this in pancreatic neuroendocrine tumors. We're measuring MGMT. Is there an optimal dose and schedule? We don't know yet. Comparisons among trials are very difficult because the eligibility criteria might be very different from one trial to the next, and how they define response might be very different from one trial to the next. So those trials I showed you with a 60% response rate down to a 9% response rate, those were using completely different response criteria, so you can't compare them. Chemo combinations with targeted agents, that's the next exciting area, I think, and that's what Pam's going to talk about. These are recent trials I won't mention. Pam will mention this one a little bit. This is Emily Bergsland's trial from UCSF. And I guess just bearing mention, this is a trial from another colleague at Moffitt Cancer Center in Florida that showed the very good response rate for pancreatic neuroendocrine tumor. And this is the only slide I have on PRRT. So just as sort of a comparison, this PRRT, this is taking a very hot radioactive isotope, linking it to octreotide or octreotide-like drug, injecting an IV, letting it concentrate in the tumor, but instead of just imaging the tumor, this time you're actually delivering enough radiation that you're trying to kill tumor cells. And so that's 500 some patients that they've treated in Rotterdam, and these are the reports. Measured in response rate, this is partial response, which is how we define response, which is partial plus complete. You can see the carcinoid, again, 1% plus 22 is 23%. Pancreas, 6% plus 36% is 42%. So again, always higher in the pancreatic neuroendocrine tumors. And so that's a pretty reasonable response rate. And you can compare with Pam's data in a second. Again, apples and oranges. But there's no doubt that that therapy can shrink tumors. So I'm sorry we took up your time that you couldn't explore the campus if you haven't already. But this is the quad with the church. It's about 2 blocks that way. And I'm going to let Pam talk about targeted therapies. And then we'll both take questions.